Okay, so now let's go to your ribonucleic acid. So your ribonucleic acids, as mentioned, yung pagkakaiba nila dun sa DNA, di ba, na-discuss na natin kanina. So ribonucleic acids are, the sugar unit is ribose, yung base niya is thymine rather than uracil, and another thing I forgot to mention pala, your DNA is a double helix, your ribonucleic acids are generally single-stranded. So pag DNA, double, pag RNA, single. Okay? So, your RNA also tend to be much smaller than your DNA molecules kasi pag gumagawa tayo ng RNA natin, we only get the portion that we need from the DNA. Tapos, you transcribe it into RNA and that's it. You don't get the whole length of the DNA molecule. Kaya mas maliliit si ribonucleic acid natin. Excuse me. So your ribonucleic acid has 6 types. The first of which is your rRNA or your ribosomal RNA. So this is the RNA that is found in your ribosomes. It's the most abundant type of RNA in our cells. So the rRNA associates with proteins in order to form your ribosomes. Okay? So next, you have your mRNA or the messenger RNA. So the messenger RNA is the one that carries information that specifies the amino acid sequence of a protein to the ribosomes. So kung meron kang DNA molecule, di ba? Sabi natin DNA is a double helix. Your DNA is transcribed into an RNA, which is your messenger RNA. So the messenger RNA is single-stranded. So that's the messenger RNA serves as the template for synthesizing your protein. So your protein, so the mass of your mRNA also actually depends on what type of protein is being produced. Siyempre, kung mas maliit na protein lang yun, mas maiksi yung mRNA. Pag mas malaki, mas complex yung protein, siyempre, mas malaking mRNA molecule yung kailangan para ma-synthesize siya. Okay? Next. You have naman your hnRNA and also known as your heterogeneous nuclear RNA or your PT or primary transcript RNA. It's also known as your pre-mRNA. So, ano yung hnRNA? So, hnRNA is an RNA that is found only on the nucleus. It is formed directly by DNA transcription. So, kapag meron kang DNA and you use it to form your RNA, so yung RNA mo actually made up of single-stranded yan, di ba? So, ribose plus nitrogenous bases, tas mahabang strand lang yan, si RNA mo. So, direct from DNA, you form your hnRNA. And then you process your hnRNA into the final form in order to produce your mRNA. And what is responsible for the processing? For the post-transcription processing, it's your snRNA. So, snRNA facilitates the conversion of hnRNA to mRNA. So, from DNA, you produce HN, and then you produce M. So, and the one that facilitates that one is your snRNA. Okay? Next, you have naman your tRNA. So, tRNA is the transfer RNA. It delivers amino acids to the ribosomes for protein synthesis. So, when you want to produce proteins from this one, the one involved, aside from the ribosomal RNA in the rRNA, of course, is the other one involved is your transfer RNA. When you want to make protein from the mRNA. Okay? And then you have molecules, RNA molecules that have enzymatic activity. Di ba sabi natin, your enzymes are mostly proteins. So, almost all of your enzymes are proteins except for some exceptions. So, ito yung exception na yun. Si ribozymes are RNA molecules that have enzymatic activity. So, we will not focus on that one. So, let's go to the process on which your RNA is transcribed, no? So, nandito na tayo sa central dogma. So, tapos na tayo sa replication. Later na natin to discuss sa clinical significance. So, nandito tayo sa conversion of DNA to RNA in the process of transcription. Okay? So, what happens during transcription? So, di ba, na-mention natin yung key differences between DNA and RNA. So, your DNA is single-stranded. Your RNA is, your DNA is double-stranded rather, and then your RNA is single-stranded. So, kapag gagawa ka ngayon ng RNA mo, isang strand lang yung gagawin mong template kasi nga single-stranded lang yung gagawin mo. So, didikit lang siya dun sa isang strand. And then pag natapos na siyang makasynthesize, aalis na siya. Hindi siya mag-stay bonded to the DNA molecule, okay? So, what happens during transcription? Syempre, when you convert your DNA to RNA, your deoxyribose becomes ribose. Naalala din natin na different yung nitrogenous bases between DNA and RNA, di ba? Sabi natin, pag DNA, you have, both of them have adenine and guanine, pero yung, and cytosine. Pero, yung thymine found only in DNA, yung uracil found only in RNA. So, yung mga thymine bases become uracil. Another key difference between the replication of your DNA and transcription is that your transcription does not need a primer. Kasi nga RNA na siya. So, hindi niya kailangan ng RNA primer. So, mag-start na siya without the need of a primer. And also that there is no proofreading involved. Hindi kagaya nung sa DNA replication na merong meron tayong way of keeping the integrity of the DNA molecule. Dito sa RNA, wala na nung ganong mechanism. Okay? So, what happens in transcription? DNA is used to synthesize hnRNA. hnRNA is processed to form mRNA. So, what happens during the processing? Ito na yung step-by-step niya. So, DNA unwinds exposing the desired sequence. And then, of course, as parehas lang doon sa kanina, free ribonucleotides, ribo, hindi deoxy, kasi RNA na yung ginagawa natin, free ribonucleotides align on one of the strands or which serves as the template strand to form your new base pairs. So, sabi natin, in RNA, you have uracil rather than thymine. So, the uracil nucleotides will align with the template strand with the adenine bases on the template strand. Okay? So, next, the one that is involved in linking this, kung sa DNA, ang involved in linking is DNA polymerase. Siyempre, dito sa RNA, you have your own polymerase. So, the RNA polymerase is involved in linking the ribonucleotides to the growing hnRNA molecule. Okay? So, and then, after the RNA polymerase encounters a stop signal, then the transcription stops and the RNA is set free. Okay? So, meron ka na ngayong hnRNA molecule. So, then the hnRNA will undergo what you call the post-transcription processing. So, your hnRNA will be converted to mRNA. So, the mRNA is the one that goes out of the nucleus and is transferred to the ribosome for protein synthesis. So, yung genes nga natin, di ba, sabi natin, everything starts from the DNA, which is converted to RNA, which is converted to protein. So, the base sequence that is desired, you call that the gene. So, your gene is a segment of the DNA. It's the base sequence. And it consists of alternating introns and exons. So, kung meron kang gene na gusto mong synthesize, gusto mong i-transcribe, you get a base sequence. And the base sequence actually is made up of two things. Yung exons, which do not code for genetic information, and the introns, the exons which code for the genetic information, and the introns which do not code. So, si introns, umenter lang yan para lang, wala lang. Okay, so, itong si introns are in between the exons. So, the exons are the one that is exhibited. So, si exons yung actual, si introns yung salingkit-kit. Okay? So, what happens in post-transcription processing? You have your splicing. So, tinatanggal yung mga introns that do not code for information. So, the introns are removed. So, when you remove the introns, you put the DNA, the exons together in order to, and when that happens, you form your mRNA. So, the process is, of course, facilitated as mentioned by your snRNA. So, yung deletion of introns and splicing of retained exons is facilitated by snRNA. And that is your post-transcription processing. Okay? So, meron din naman tayo nung tinatawag nating alternative splicing. So, actually, nung nag-Human Genome Project, when they wanted to map out the entire gene, all the set of genes found in a human body, na-realize nila na hindi pala ganun kadami yung genes as estimated. Kasi ine-expect nila nun, if you have a protein, you have a certain gene that codes for it. Pero So, ine-expect nila na yung number of genes natin is in the 100,000s. Kasi yung number of proteins sa katawan natin is in the 100,000s. Pero, na-realize nila na kahit na 100,000s yung proteins natin sa katawan, yung number of genes natin is actually around 20, 30, or 40,000 lang. So, sabi nila, bakit ganon? Bakit ang konti lang ng genes pero ang daming klase ng proteins na natatagpuan sa katawan? So, one explanation for that is your alternative splicing. So in alternatives, yung sa ito natin, in the body, the estimated number of proteins vastly exceeds the estimated number of genes. So the alternative splicing is the process by which several different proteins that are variations of a basic structural motif can be produced from a single gene. So ano yung sinasabi nun? Meron kang isang gene lang, tapos from that single gene, pwede kang gumawa ng iba't ibang klase ng protein during the process of alternative splicing. Okay? So, uh, So, ito sa picture na to, makikita niyo, magkakaroon tayo ng idea kung ano nangyari sa alternative splicing, no? So, for example, you have your first protein. You want to come up with protein that is uh, yellow, pink, and orange. Okay, so yellow, pink, and orange, you want that. Pero ito yung HNRNA molecule mo. So, what happens during alternative splicing? You splice this intron, you splice this intron and the exon in between. So, pag tinanggal mo itong exon na to, itong intron section na to, and then this intron, you come up with yellow, pink, and orange. So, you come up with the first protein. Okay? Next, another way to transcribe from, to come up with, to transcribe from, to get another type of mRNA is by splicing this HNRNA in another way. So, for example, you want to come up with this second protein, yellow, green, and orange. So, gusto mo, ang material lang is yellow, green, and orange. So, of course, uh, you remove this entire section. You remove this intron. So, you get yellow, green, and orange. So, you get another type of protein from the same HNRNA. Okay? Next, you want this third protein naman. So what you do, you remove all the introns and then you put them together, put the remaining parts exons together, you get the third mRNA. The mRNA for the third protein. Gets? And then for the last one naman, gusto mo lang is yellow and orange. So what do? You remove this entire section of introns and the exons in between. So, dalawa lang yung natira, naging yellow and orange lang, and you get a fourth type of protein. So, ganun yung nangyayari sa alternative splicing. You one whole segment, you have a segment of gene, you have a segment of HNRNA, and then you cut it in different manners. And then you put them together, the parts that you want, so that you get a certain type of protein that you want. Okay? So sabi natin, meron tayong human genome. So, genome is the set of all genetic information, di ba? So, meron din naman tayong tinatawag na transcriptome. So, the transcriptome is the set of all mRNA molecules that can be generated from a genome. So, syempre, mas malaki to kasi nga, meron tayong alternative splicing. This is much larger than the human genome. The human transcriptome is many times larger. Okay? So, next. So, summary lang to. Kung ano nangyari, di ba? DNA, during transcription becomes HNRNA, heterogeneous nuclear RNA. When it undergoes splicing using snRNA, you form your mRNA. And then your mRNA is converted into proteins. So yan, di ba? So you have this. Pag nag-alternative splicing ka, you can form many different combinations. So may different proteins. So I just like this picture, so I added it here. Pero same lang yan. Okay. So now let's go to translation. So during translation, if you remember our central dogma, translation is the process by which you convert RNA to protein. So bakit naging translation yung tawag sa kanya? Kasi para kang nag-convert into another language. Kasi yung language, di ba, ng DNA to RNA, parehas lang silang talking about nitrogenous bases. So, nitrogenous base, adenine, thymine, base pairing. Parehas lang sila halos at pinakaiba lang nila yung sa uracil, yung sa thymine. Dito, kay RNA at kay protein, meron siyang sariling code na sinusunod. So, dito sa translation, you follow what you call the genetic code. So, what is the genetic code? It's the nucleotide C. So, ito yung code na sinusunod when you convert your RNA to protein. So remember, the nucleotide sequence in a given mRNA determines the amino acid sequence for the protein being synthesized. So kung ano yung mRNA, yun yung gagawin. Yung mRNA, nandun sa kanya yung instructions kung aling amino acids yung kailangan mo para mag-produce ng certain protein. So dito sa genetic code, ang ginagawa is when you make your proteins, you read the ribosome actually reads 3 base sequences. So combination siya ng tatlong nitrogenous base ang ginagamit to specify an amino acid. So the 3 base sequence used to specify an amino acid is a codon. Okay? So ilan ng bases natin, 'di ba? We have 4 bases. So AUG. So AUG and C. So apat yung bases natin. So for 4 bases, you can make 64 possible codons. So, kasi 3 base sequence, di ba? So, 4 raised to the 3. 4 times 4, 16 times 4, that's 64. So, 64 possible codons for each specific amino acid. So, 20 lang yung amino acids natin. So, may nauulit. Okay. So, you have 64 possible codons kasi you have 64 possible 3 base sequences. 61 of which codes for a specific amino acid, 3 of which codes for stop signals. So dun sa 64 na possible codons, merong 61 of them amino acid, 3 of them stop signal. Tapos dun sa 61, isa dun is the start codon. So, the start codon is AUG. Tandaan nyo to, this is very important. So, the start codon is AUG, which also codes for the amino acid methionine. Okay? So, methionine, naalala nyo may sulfur to, diba? Methionine sa pangalan. So, AUG is the start codon. It codes for methionine. So, pag may nakita siyang AUG, for example, mo RNA ka, tapos siya ribosome, pag may nakita siyang AUG, it's the start part. So, mag-start na yung transcription. So, starting signal is AUG, the start codon. Okay? If naman, it goes along translating, tapos may na-encounter siya ng any of the stop codons, the translation will terminate. So, what are the stop codons? It's UAG, UAA, and UGA. So, how do you remember the stop codons? Kasi mas mahirap siyang tandaan kaysa ka AUG. Sa AUG, isa lang. So, AUG, start codon. Pag stop codon naman, you want to stop the baby from crying. So, wag, uwa, uga. Diba? Yung wa is the crying baby. Wag mo siyang ugain. So, wag, uwa, uga. That's the stop codon. Okay. So, next. So, what are the properties? So, ano muna yung mga codons na sinasabi natin? Diba? Sabi natin, merong 3 genetic, meron tayong 3 base sequences, diba? So sabi natin, each 3 base sequence will encode for a specific amino acids, except for 3 na magko-code ng stop codon, diba? So, for example, here, you have a 3 base sequence, para mo babasahin tong table na to. The first base is U, the second base is another U, and the last base is another U. So, what amino acid do you form? You form UUU, that's phenylalanine. So, pag may nakita kang UUU, ang i-encode niya is phenylalanine. If you form for example, your first letter is letter C, the second letter is letter A, and the third letter is letter A. So, ano mo form mo? So, CAA, that one is glutamine. So, ganun siya basahin. Ito yung guide natin kung paano i-translate from DNA to RNA. Kaya ka siya naging translation kasi para kang nagta-translate from one language to another, from the language of the DNA base pairing, the DNA RNA, from the language of the RNA base sequence, i-convert mo siya into codons, 'di ba? So yung codons na yun yung ginagawang amino acids, okay? So there are several characteristics of your genetic code. So the first one is that the code is unambiguous. Ano ibig sabihin ng unambiguous? For each codon, for each combination, it only specifies one amino acid. Hindi siya nagsispecify ng other amino acids. What do I mean by that? For example, you have a code here. So, kunyari ito, si GCU. GCU will only ever code for alanine. It will not give rise to aspartic acid. It will not give rise to glycine. It will only ever code for alanine. Yun yung ibig sabihin ng the code is unambiguous. Another thing that you have to remember is that the code is degenerate. Bakit sinabi natin degenerate or redundant yung code? So, sabi natin, si GCU will only ever code for alanine. Pero, there are other codons that can also code for alanine. Kaya siya naging redundant or degenerate. Kasi GCG can also code for alanine. GCA can also code for alanine. GCC can also code for alanine. Okay? So, tandaan natin, it's degenerate because more than one codon can code for one amino acid. But remember that each codon can only encode for one amino acid. Okay? Nakalito ba? Ulitin natin. So, each codon can only encode one amino acid. GCU will only ever encode for alanine. ACU will only ever encode threonine. AAU will only ever encode asparagine. Hindi siya magbibigay ng ibang klaseng amino acid pag yan yung sequence mo. Okay? However, the code is also degenerate in that you have multiple codons that can code for the same amino acid. So, you have multiple codons that can encode for the same amino acid, like this one. So, AAA and AAG can both code for lysine. So, bakit ganon? Bakit itong si valine, 4 na possible codons niya? Bakit si phenylalanine, 2 yung possible codons niya? Bakit si cysteine, 2 lang? Bakit si serine, 2 lang? Bakit si tryptophan nga, 1 lang eh. Sabi nila, dahil daw to sa relative amounts of the amino acids found in our body. So, pag mas marami daw tayong valine sa katawan kaysa sa tryptophan, for example. Kaya, mas maraming codons that code for certain amino acids. Okay? Next, another thing about the genetic code is that the code is universal. So, bakit siya naging universal? Kasi yung genetic code mo in bacteria, for example, kung ganito yung sequence ng mRNA doon, ng RNA doon, and then yung code mo sa hayop, ganito yung sequence, yung CUU sa hayop will code for leucine, yung CUU sa tao will code for leucine, yung CUU sa fungi will only code for leucine. Ganun. It's universal. It's the same in all organisms. Okay? So, next. Another thing is that the code is comma-less. So, walang spacers or commas. Tuloy-tuloy lang yung pagbabasa sa kanya. So, first 3 is a codon. The next 3 is a codon. The next 3 is a codon. The next 3 is a codon. So, tuloy-tuloy siya binabasa ng ribosomes natin when they make proteins. That's why, very important na hindi magkamali. Kasi, kapag nagkaroon ng mali doon, kunyari may natanggal na isa, or na magbabago na yung protein na nasi-synthesize. So, yun. So, here are the rules that I mentioned. The code is unambiguous. Each codon specifies no more than one amino acid. Okay? The code is degenerate or redundant. More than one codon can specify a single amino acid. And another thing that I forgot to mention, amino acids with more than one codon that are coded for by more than one codon, the first 2 bases are usually the same. So, dito, di ba, makikita mo. So, si leucine, dalawa yung codon that codes for it, it's UUA and then UUG. So, the first 2 bases are the same. So, ganun din dito. Si tyrosine, UAU, UAC. So, AGU, AGC. Ito nga, sa AG, puro sila GC, and then UCAG, they're all alanine. Okay? Gets. And then the last one, not the last one. The code is universal. The code is the same in all organisms, and the code is comma-less, and there are no spacers or commas between codons. So, hindi siya nag-stop, tuloy-tuloy lang yung basa sa kanya. Okay? So, let's have some examples dito sa codons natin. No? So, how do you use the genetic code to predict the amino acid sequence? So, lakihan natin yung picture para mas madali natin siya makita. Okay? Where is my annotation? Okay. So, use the genetic code to predict the amino acid sequence. So, you have a GCC here. So, ano ang magiging product nun? You have a G, you have a C, and then you have another C. So, what is GCC? That's alanine. So, this one is ala. You have an AUG. Alam niyo na naman yung AUG. Sinabi natin, AUG is the start codon. It also codes for methionine. So, this is ala. This one is met. And then you have your GUA. So, G, U, and then your A. So GUA, that's valine. So that's valine, ala, met, val, and then 3 A's, A, and then A, that's lysine. So this one is lyse. And next you have your U, G, and then C. So UGC is cysteine. So cis, and then G, A, and C is aspartic acid. And then you have your C, and A. So CCA is proline. So yung amino acid sequence na ito, you can predict it from the using the genetic code, diba? So this one is alamet, val, lys, cis, asp, and pro. So ganyan yung sagot dyan. So madali lang siya. Okay? Next. So how does translation occur? The one that meron tayong, di ba binanggit na natin to? Highlight lang natin yung tRNA molecule. So, sabi natin kasi, the one that carries the amino acids to the ribosome is your tRNA molecule. So, your tRNA molecules function as intermediaries that deliver amino acids to the mRNA and ribosome for protein synthesis. So, may parts yung tRNA natin. No? Ito yan. You have the 3' end, you have the anticodon loop, and the anticodon. So, ano nangyayari dito? You have this is the RNA molecule, tRNA molecule para siyang clover leaf. So, para siyang, meron siyang hairpin loops. So, meron siyang loops dito, meron siyang loop dito, and then it has a loop over here. And in this loop, meron kang 7 amino acids. The middle 3 is what you call the anticodon. So, you have a 3' end. So, di ba sabi natin, 5' to 3' ang directionality ng nucleic acid sa atin. So, meron kang 5' and the 3' end here. So, single-stranded siya, pero it folded upon itself. Kaya nagkaroon ng bonding dito sa pagitan. So, you have 5' end, and then your 3' end. Sa 3' end, dun uma-attach yung amino acid that is being delivered. And then, dito, sabi natin, you have the anticodon loop on the opposite side. Yung anticodon loop has 7 paired bases. So, the 7 paired bases na yun, they have 3 of which 3 of the bases is called the anticodon. So the anticodon is the 3 nucleotide sequence on the tRNA molecule that is complementary to a codon on an mRNA. Kasi di ba may mRNA strand ka? Tapos yung 3 base sequence mo is you call that a codon. So in order to get the appropriate amino acid that corresponds to that codon, kailangan yung tRNA mo meron siyang anticodon loop na complementary to this codon para yung amino acid that corresponds to this codon is the one that will bind to this tRNA molecule. Okay? So it will correspond to the codon, not the anticodon. So this one corresponds to this one. Okay? Next. So, doon na tayo sa process of translation. So, during translation, your translation is started during initiation, wherein your mRNA molecule will bind to the small subunit of the ribosome. So, sa small moon, hindi pa siya buong subunit, hindi pa siya buong ribosome. So diba sabi natin during the cell discussion, you have a small subunit and a large subunit in your ribosome. So, yung mRNA will attach to the small subunit, and then, it will align itself in such a way that the start codon will align to something called the P site or the peptidyl site. So, the AUG or the start codon will align at the P site. And then, what happens is that an activated tRNA, pag sinabing activated, yung meron na siya naka-attach na amino acid, will bind to the codon on the P site. So, syempre, yung activated tRNA na yun, kailangan, ang anticodon niya is complementary to the AUG or the start codon. So, syempre, yung complement is UAC. So, yung tRNA with the UAC, with the complementary anticodon, carrying the methionine which corresponds to AUG, siya yung magbabind dun sa P site. Okay, so the activated tRNA with the complementary anticodon attaches to the start codon by complementary base pairing. So, complementary base pairing yung nag-hold together dito. So, ang meron ka pa lang here at this step, you have the small subunit and the RNA and the tRNA. When the large subunit binds, you form what you call the initiation complex. So initiation complex is made up of the mRNA, the small subunit, the large subunit, and the tRNA. Okay? So, tandaan sa initiation, your start codon, your AUG, is on the P site. Okay? Next. So, next to your P site, you have your A site. So, this is the P site. And then, the next one to that is your A site. So, ang nangyayari dito sa elongation naman, the site adjacent or the A site, the A site codon will be aligned to the syempre, yung next code, syempre, yung sabi natin kanina, yung AUG is on the P site. So, yung next codon sa kanya, nandun siya sa site na katabi niya. So, yung start codon is on the P site. Then, the next codon is on the A site. So, the A site codon, of course, is bonded to by a tRNA with the matching anticodon. So, for example, here, the matching anticodon corresponds is on the tRNA and the amino acid is over here, si glycine. Okay? So, you have a met and then you have a gly. So, what happens is that peptidyl transferase will link this P site amino acid to the A site amino acid. So, ang mangyayari is actually, yung met na to, aalis siya from binding dito sa tRNA na una, lilipat siya, didikit siya, kaya siya naging transferase kasi the met will transfer to this amino acid. Will transfer binding to the next amino acid. So, didikit siya doon sa amino acid on the A site. So, kaya siya naging peptidyl transferase. So, the peptidyl will be transferred to this one. Okay, so the tRNA at the P site, dahil wala na yung met na hawak niya, will become free and then it will leave. So, excuse me. So, aalis na yung tRNA na dahil wala na siyang laman. And then, what happens is the ribosome will move along the molecule. So, pag galaw ni ribosome, gagalaw siya this way, in such a way na yung kaninang A site mo magiging, yung kaninang codon mo na nasa A site, yun ang magiging codon mo na ngayon sa P site. So, tatapat na siya sa P site ngayon, yung second. Okay? So, next. So, dahil nandun sa second, nandun na sa second codon tayo, no? So, dahil nag-move na si ribosome mo, syempre, nakadikit sa kanya yung dipeptide na nagawa mo nung una. Okay? And then, since meron ka na ulit codon dito sa A site na becomes empty kasi ginalaw mo na yung ribosome mo, may magbabind na naman dyan na bagong tRNA carrying another amino acid that corresponds to the one on the codon. Okay, so magbabind ulit yung bagong tRNA and then this dipeptide will be transferred by peptidyl transferase onto the amino acid at the A site. Okay, so ganun yung paulit-ulit na nangyari until mabuo mo yung peptide molecule. So, let's walk through what the statement is saying, no? So, the site adjacent to the P site is called the A site. So, this is the P site and this is the A site. And then, the A site codon is bonded and exposed and bonded to by a tRNA with the matching anticodon. So, ito yung anticodon na matching. Tapos, and dito yung amino acid na kailangan niya. And then, the peptidyl transferase will link the P site amino acid with the A site amino acid to form a dipeptide. So, magiging dipeptide na siya on the T site, on the A site tRNA. Okay? And then, the tRNA becomes free and is released. So, na-release na siya and the ribosome will move. So, gagalaw na yung ribosome. So, ito, pag gumalaw na siya, syempre, nandito na yung AUG. Nandito na ngayon yung second codon mo sa P site. Imbes na nasa A site siya kanina, na si second codon mo is now on the P site and then the next codon is free. So, the next codon, the third codon, will now be on the A site, which means pwede na mag-bind yung bagong tRNA that carries the corresponding amino acid. Okay? Next. So, the shifting is also called translocation. So, yung next naman, syempre, may nag-bind na ng bagong tRNA with a peptide that will So the tRNA will the peptide will move to the A site, the P site, tapos may expose ka na codon, and then may papasok ulit na tRNA. So the codon at the A site bonds matching tRNA, and the peptide at P site is transferred. So ito na naman dipeptide mo will transfer to the peptide to the amino acid here. So in order to form a tripeptide. Okay? So, paulit-ulit lang yan, paulit-ulit hanggang maubos mo yung length of the mRNA mo, matapos mo yung pagbawa ng protein. Okay? So, what happens during elongation? This is just a closer look at the reaction. What happens is that the peptidyl transferase enzyme catalyzes the acyl transfer reaction. So, anong nangyari? For example, this is the start codon, di ba? Si AUG, start codon, so this is for starting reaction. You have an acyl group here. So, this one will attach to the glycine. So, this C will bind to this. So, magiging ganyan siya. And then, once this one is over, so pag meron ka na ulit bagong tRNA dito, if you have another tRNA here on the next site, then this one will transfer to the one on that. So, pahaba siya ng pahaba. Magagawa ka ngayon ng peptide chain. Okay? So, tuloy-tuloy siya. When does it stop? It stops when a stop codon is reached. Si wag, si uwa, si uga. Okay, so any of the stop codons can terminate the process and the polypeptide is cleaved from the tRNA via a hydrolysis reaction. So, you add the water, tapos ma-break off siya. Tapos you have your free protein molecule. So, of course, once the translation is over, the ribosome will dissociate and the mRNA will become released. Okay? So, after termination, after your translation, you have something you call the post-translation processing. So, ito yung finishing touches on the proteins that we synthesize. So, it gives the proteins its final form before it becomes completely functional. So, ano yung post-translation processing natin? There's several examples of, the first one is removal of the methionine residue during initiation. Kasi diba sabi natin the start codon codes for methionine. Pero hindi naman kasi lahat ng protein natin kailangan nagsa-start sa methionine. Hindi lahat ng protein natin nagsa-start sa methionine. So, bakit? Kasi tinatanggal in some cases, tinatanggal nila yung methionine residue via hydrolysis para wala siyang methionine. Okay? Another means of post-translation processing is covalent modification. For example, you form disulfide bridges within your cysteine residues. Sinabi natin ito sa protein synthesis. And then, another example is completion of folding para magawa na yung desired shape ng molecule natin. For example, you want to make an enzyme, etc. Siyempre, you complete the folding of the polypeptide in order to make the final protein that will become functional. Okay? So, here is an overview na video on how translation works. Dito sa video na to, meron siyang extra na site na sinabi, which is the E site. Pero, E just actually stands for exit. Okay? So, hindi siya ganun ka-relevant actually. Hindi naman siya super important. So, madalas, kahit dun sa textbooks, meron din E-site, yung iba naman, ina-omit na nila kasi it's basically just moving out of the way. Okay? So, let's go here. Let's watch this video. How do I enlarge this? Para maging mas malaki siya. So, lakihan natin. Para mas kita niya. Okay.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Okay, so that's the link for the video if you want to watch it again. So here, you have something you call your polysomes. So polysomes or polyribosomes. This describes how efficient our cells are at making proteins. Kasi imagine mo kung meron kang gagawa ka ng maraming protein, maraming copies of a certain protein. Tapos isang mRNA lang meron ka. So, if you need to make an mRNA for each time you make the protein, masyado siyang tedious, masyado siyang it will consume too much resources. So what the body does is actually, it makes your uh, polysomes or polyribosomes. So, what happens is you have an mRNA, a complex of mRNA with several ribosomes. So, that's your polysome. So, ano nangyayari dito sa polysome? So, here using your polysomes, you can, identical protein chains can be synthesized almost simultaneously from one strand of RNA. So, ano nangyayari dito? As the ribosome moves along the length of the mRNA, another ribosome can bind to the place it vacated. So, ano nangyari? Di ba sabi natin, when you have an mRNA, gumagalaw siya, nagmo-move siya along the molecule as you read the mRNA. So, for example, you have this, molecule, di ba? You have this ribosome, you have this complex. So, meron kang protein dyan. So, when it's done reading whatever codon is there to read, gagalaw siya, magta-translocate siya, and magmo-move siya. So, what happens during polysome formation, since gumalaw na siya, another ribosome will go to the place kung saan siya nanggaling. Okay? Tapos, magsa-start na siya ng synthesis over there. So, since gumagalaw yung mRNA mo, itong ribosome na to, lumipat na siya dito sa kabila. Diba? Tapos, meron ka ng bago dito. And then, pag gumalaw na to ulit, may pumasok ulit na ribosome dito. And then, when this one moves to this one can move to this one kasi, di ba, it's a, para sa conveyor belt na dumadaan. So, for example, you have this ribosome. Pag gumalaw siya, pwede nang may mag-bind dito. Okay? Ganon. Okay? So, you have a ribosome here. When it moves, delete. Okay, sorry. You have a ribosome. When it moves to become this one, this place becomes empty. So, pag naging empty na tong unang part, then you can actually have another ribosome binding to that area. So, may papasok na ulit na ribosome dyan. Okay? So, magkakaroon ka ng ribosome over here. When this moves, this one will also move. Okay? Gets? So, pag nag-move na sila parehas, nag-move na sila parehas, then this becomes empty. So, meron na ulit, pwede mag-bind na bagong ribosome dyan. Gets? And when this moves, to encode the next codon, this one will move to this one. And then, yung nandito, lilipat siya, magiging ganito. And then, it will become empty. And then, you can put another ribosome there. Para tuloy-tuloy na gumagawa siya ng maraming copies of the same protein. Okay? So, summary ng transcription and translation, which we just discussed. So, transcription is the conversion of your DNA to RNA. How do you do that? Your DNA is converted to RNA. You first form your hnRNA using your RNA polymerase, no? So, when you form your hnRNA, meron pa siyang introns and exons. When you remove the introns from the hnRNA molecule, you form your mRNA. So, the mRNA then leaves the nucleus and enters the cytoplasm in order to proceed to protein synthesis. So, sa protein synthesis, the first step, of course, is activation of the RNA kung saan didikit yung appropriate amino acids, appropriate to the codon that is being looked at. So, for example, here, yung AUC, the appropriate codon for that is AUG. So, ang amino acid dito sa tRNA na to is methionine. Okay? So, then, you start the initiation complex. Sa initiation complex, the tRNA will bind to the P site, and then another tRNA will bind to the A site. Tapos yung amino acid on the P site will transfer to the tRNA on the A site. Tapos the ribosome will move, and then another tRNA with amino acid can bind to the A site. Tapos the peptide chain, the growing peptide chain will transfer to the amino acid on the A site. So paulit-ulit siya nangyari until the peptide chain becomes longer until you hit a stop codon. And when you hit the stop codon, there will be termination of the translation, and the peptide chain can undergo post-translation processing. So to finalize the form of your protein. Okay? So tapos na tayo sa replication, tapos na tayo sa transcription, tapos na tayo sa translation. So, doon naman tayo sa mga medical correlations para naman medyo interesting yung discussion natin. Hindi lang tayo puro bago. So, some of the medical correlations are actually pag merong error during the process of replicating your DNA. So, if you have an error in the base sequence, if a gene reproduced during DNA replication, pag nagka-error ka kasi sa pagkopya na yun, pwede mo siyang mapasa during transcription. So, dahil mali na yung kinokopyahan mo, mali na yung mga kopya, during transcription, mali na yung matatranslate. Mali na yung magiging proof. So, it's a chain reaction. So, para kang naglaro ng pass the message, di ba? Tapos, nagkamali ng sinabi yung isa. So, dahil mali na yung sinabi niya, mali na yung maririnig nung kasunod. And the last person, probably, iba na yung message na magiging intindi niya sa message na pinasa nung unang tao, di ba? So parang pass the message, pero nagkamali yung isa. Okay. So, there's several types of mutations. You have a silent mutation. So, sa silent mutation, so this one is the normal. Mas madali siya makita kasi kung may example tayo. So, you have a silent mutation. This one is a normal, di ba? So, the normal sequence is met ala isoleucine ARG. Penilalanine, leucine, proline, isoleucine, and glycine. So, kunyari ito yung DNA mo, and then this is the amino acid that is being encoded. So, in silent mutation, nagkaroon ka ng mutation. Pero bakit siya naging silent? So, dito sa example, nagkaroon ka ng mutation dito sa letter A, dito sa leucine na to. Okay? Imbis na TTA, naging TTG. Pero bakit siya naging silent? Kasi TTA and TTG both encode for leucine. So, kahit na nagkamali siya, okay lang. Kasi yung leucine mo is encodes for the same thing. Your DNA encodes for the same thing. So, yung resulting amino acid niya is parehas lang. So, this one is more harmless than the others, of course. Kasi, kahit na nagkamali, forgivable. Kasi, the end product is the same. Okay? Next. Another type of mutation is the missense mutation. Ano naman yung missense mutation? Pag missense mutation, ibig sabihin, nag-iba yung gusto niya ipahiwatig. For example, here, ang nangyari dito, you have your TTA, doon ulit tayo sa leucine molecule, ay doon din tayo ulit sa leucine na codon, sa codon that codes for leucine. So, TTA, imbis sa TTA, you form a TCA. So, dahil nagkaiba, yung second, naging TCA rather than TTA. Instead of coding for leucine, you code a serine. So, ganito yung nangyari. Nag-iba yung amino acid. So, pag nag-iba na yung amino acid na yan, edi magiging iba na yung protein, magiging iba na yung structure ng protein mo kasi nag-iiba na yung amino acid involved. Okay? Next. You have your nonsense mutation. So, kanina missense, this one is nonsense. Ano nangyari naman dito? Kapag yung DNA mo, nag-mutate in such a way na ang na-encode niya is a stop codon. So, for example here, di ba yung DNA mo, originally it's TTA, and then it becomes a TGA. So sabi natin pag mRNA, di ba? When this one is converted to mRNA, magiging UAG. And UAG is a stop codon. So when the DNA coding strand encodes for a stop codon, ang tawag sa mutation na yun is wag. Kasi magiging nonsense siya kasi magiging putol yung amino acid na mapoproduce mo kasi meron kang biglang stop codon. Imbes na dapat meron ka pang amino acid sequence here. Okay. So this one results in a shorter mRNA and a shorter amino acid. Okay? So next, you have naman what you call a frame shift mutation. So ano nangyayari sa frame shift mutation? For example here, the original one is TA, dito kay phenylalanine. Ang nangyari is naging TAC, TAT. What happens here, di ba? This one sequence parehas lang naman dito. So ano nangyari? Nagkaroon ng deletion or nabura yung isang, natanggal yung isang letter T. So ano nangyari? So natanggal tong isang T na to, lahat ng to, syempre, ang base sa kanya, lilipat siya pag ganun. So, ang base sa kanya, nagkaroon ng frame shift, nagkaroon ng shift in the reading frame of the codon. Diba sabi nga natin, the genetic code is commaless. So, pag natanggal yung isa, matanggalan ka ng isang ng isang base dyan, lahat nung nasa kanan, gagalaw silang lahat. So, lahat sila, gagalaw silang lahat. So, ang magiging, imbis na ang basa mo, dito is like, di ba may TTA ka dyan, and then CTT, ATA, GGG, magiging, dahil natanggal na to, ang magiging basa mo is TAC, CTA, TAG, GG, whatever. So, ganun yung nangyari dito. Nagkaroon ng frame shift dahil kulang ka ng isang T. So, ganun yung frame shift mutation. So, sa kanilang lahat, ang pinakamalaking effect actually is your frame shift mutation. Kasi, dito, pag nagkamali ka dito, for example, sa silent or sa missense mutation, isang amino acid lang yung affected. Sa nonsense din, malaki din yung effect kasi matitigil yung synthesis. Dito, you produce something that is entirely different. So, pwedeng maging malaki yung effect ng nonsense na molecule na yun. Okay? So, some examples of mutations para na may idea tayo. So, ang nangyari dito, for example, you have your sickle cell disease, which is a missense mutation. Bakit siya naging missense mutation? And how it affects the lives of people. So ang mutation dito sa sickle cell disease is on the 6th amino acid. Instead of having a valine, instead of glutamic acid, you have a valine. Sorry, baliktad na naman yung pagkakatype ko. 6 global nga pala to, hindi siya glutamic. Glutamic acid becomes valine. So, 6. Sorry. So, your 6 global, so si sickle cell disease, dito, as you can see here, the DNA sequence is normally CTC. Okay? The complementary is GAG. Okay? So, ano nangyari? Dito, nagkaroon ng mutation. Okay? Imbis na CTC, naging CAC siya, naging GUG. So, dahil sa mutation at this portion of the genetic code, ang nangyari is, instead of encoding for a glutamic acid, nagkaroon ka ng valine. So, ano nangyari sa mga taong may sickle cell mutation? Because of the improper folding. Yung the red blood cells nila, they form sickling. Lalo na, nagkakaroon sila ng sickling or parang sickle, yung pinapang kaya sickle, yung pinapang harvest, di ba, ng mga crops. So, sickle cell, kasi it sickles yung red blood cells mo, nagiging hindi na siya discoid, nagiging ano na siya, nagiging para siyang sickle yung shape niya, lalo na pag low oxygen tension. So, syempre, this is very painful kasi imagine mo, ganyan yung dumadaan sa, sa, sa mga vessels mo, no? And it's also, it has tendency to rupture. So, mas marupok siya kaysa sa normal. Okay? So, next, you have your cystic fibrosis. So, cystic fibrosis naman, yung mga taong to, meron silang, yung mga secretions nila is actually more malapot than normal. For example, yung plema nila sobrang lapot, para siyang solid. Kung meron man silang secretions from their pancreas or anything, nagiging very dehydrated siya. Kulang sila ng tubig kasi meron silang channel error sa kanila. So, sa cystic fibrosis, meron silang mutation na meron silang frame shift mutation. So, ang nangyari dito kay frame shift, kanina yung deletion natin, isa lang, diba? Pero dito, tatlo yung na-delete. Pero, naging frame shift siya kasi hindi isang buong codon yung na-delete. So, ang nangyari is they deleted 3 amino acids here. So, ang nangyari, ang naging reading frame niya ngayon is naging this one. So, may na-delete na tatlo dito, nag-iba yung reading frame. So, dahil di pa rin to ATT, so tama pa rin sa isoleucine, pero ito na yung naging kasunod, GGT, GGT, etc. So, nagkaroon ng deletion. So, ano nangyari? Nagkaroon ng error in making the chloride channels of this cystic fibrosis patients. Kaya ang nangyari is their secretions become abnormally thick. Kaya usually prone sila sa pneumonia kasi yung secretions ng baga nila sobrang kapal. They're also prone to uh, have digestion problems, pancreatitis, etc. Kasi nga sobrang kapal ng secretions kahit sa digestive tract, etc. Sobrang lapot ng mucus dahil sa kanilang dahil sa defective chloride channels found in the cystic fibrosis patients. Okay? So next. Doon naman tayo sa tinatawag nating viruses. Diba? Sabi natin, viruses are small particles that contain DNA and RNA surrounded by a protein. And they cannot reproduce without the aid of a host cell. So how do viruses reproduce then? They invade the host cell. They invade the host cell and utilize the host cell's machinery in order to reproduce. So, pag ginagawa nila yun, usually, they disrupt the normal cell operation. Some of them even cause the cell to lyse. That's why you have pathology when your cell is invaded by a virus. Okay? So, there are two types of viruses. Merong viruses that are made up of DNA, na DNA, yung nucleic acid nila, yung genetic material nila. There are viruses na RNA, yung genetic material nila. So, pag DNA virus, syempre, gagamit sila ng DNA polymerase to convert it to RNA to make the viral protein. Pag RNA virus naman, meron two ways that it can replicate. You can use the RNA of the genetic material and then convert it to protein agad using RNA-dependent RNA. You can replicate the RNA using RNA-dependent RNA polymerase and then you convert the RNA to protein agad. Or, or you have RNA-dependent DNA synthesis using reverse transcriptase. So, ano nangyayari dito? You have an RNA virus. So, yung virus mo, yung genetic material niya is an RNA. What it does is it converts it into a DNA molecule. Tapos, pagkatapos siya mag-DNA molecule, it will follow along the sequence of the central dogma. Yung DNA template niya na yun, gagawing RNA, tapos yung RNA magiging template for the viral protein. So, ganito yung nangyayari actually in your HIV. So, sa HIV, di ba? Your HIV, your human immunodeficiency virus is called a retrovirus. Bakit retrovirus? Kasi bumabalik siya. Retro. Di ba pag retro, yung mga throwback, ganun. So, retrovirus kasi it goes back along the central dogma. From the RNA, it becomes DNA. And then, from the DNA template, it forms your RNA, your viral RNA and protein. So, ganun yung nangyayari sa retroviruses. Okay? Next. So, ito naman, another interesting topic regarding your viruses, di ba? So, yung vaccines are preparations containing an inactive or weakened form of a virus or bacterium. So, hindi lang viruses ang may bakuna, di ba? Meron din tayo yung Bacillus calmette-guerin, yung BCG, yung vaccine for TB. So, meron din tayong mga vaccines for bacteria. So, di ba? So, yung TB vaccine, it actually doesn't prevent TB. It just helps you avoid extrapulmonary TB. So, pwede ka pa rin magka-TB, pero hindi siya kakalat to the other parts of the body. Okay. So, your vaccines trigger antibody production para magkaroon ka ng, so parang in-expose ka niya so it triggers your body to make antibodies. So, para ma-recognize yun to confirm immunity against a certain disease. So, naalala niyo to, itong picture na to, di ba? Ito yung nangyari during COVID pandemic, di ba? Lahat ng tao, naka-face shield, tapos, the majority of places required you to have a vaccine certificate. So, everyone went to get their vaccines during the Res Bakuna era. So, di ba, they had several different types of vaccines during COVID. So, the first one is Sinovac. So, di ba si Sinovac is actually an inactivated vaccine. So, si Sinovac, si Sinopharm, and then the Bharat Biotech na wala naman dito sa Pilipinas, contains killed versions of the SARS-CoV virus. So, dahil patay na yun, tapos itinurok sa katawan, so, hindi naman na siya pathogenic dahil patay na siya. So, pero dahil it still contains the virus itself, patay nga lang. The immune system will recognize that and and it will trigger an immune response without causing illness. Kasi nga, hindi na siya makapag-replicate kasi patay na siya. So, this response builds your memory so your body can fight off COVID in the future. Okay? So, your SARS-CoV-2 or the COVID virus in the future. So, ano yung nangyari? So, you have a killed virus, killed vaccine, ganun yung nangyari. So, you have a killed virus, tapos na-expose yung immune system, your immune system recognizes that, and then you can, in the future, when you get exposed to COVID, you can fight it off. So, yun yung idea during ng inactivated viruses. So, marami tayong other vaccines na. Ginawa ko lang sa COVID para medyo relevant. Okay? So, another example of vaccines that we encountered is your viral vector vaccines naman. Ito naman yung AstraZeneca, as well as yung Janssen, pati yung Gamaleya or Sputnik, di ba? So, ang nangyari naman dito, they use another virus, not the virus itself. They use another virus, tapos ang laman nun is SARS-CoV-2 genetic material. So, yung genetic material ng SARS-CoV-2, pinasok siya sa loob ng ibang virus. Okay? So, ang gagawin ng cells natin pag pinasok na to, is that our cells will use the genetic material it encountered to make a specific SARS-CoV-2 protein. So, dahil iba yung virus, hindi siya pathogenic, harmless yung virus, pero meron kang kopya ng genetic material para gumawa ng SARS-CoV-2 protein. And then, the proteins will be recognized by your immune system in order to trigger an immune response so that you can fight off SARS-CoV-2 viruses in the future. So, ganun yung idea during the other types of COVID vaccines natin. And then the last one is the new one, and this one is more relevant to our discussion, is your nucleic acid vaccines or yung mga mRNA na vaccines, di ba? So anong nangyari sa mRNA vaccines? Examples of which are your Pfizer and your Moderna. What you have is a segment of the SARS-CoV-2 genetic material. So it can be DNA or RNA basta it codes for the SARS-CoV-2 genetic material dun sa vaccine natin, okay? And then, thus our cells will use this genetic material to encode for a SARS-CoV-2 protein. So, which will be recognized by the body in order to trigger the immune response. So, of course, ganun din. So, iba-iba lang sila, di ba? So, dito, mRNA, usually, yung ginagamit, di ba? So, mRNA, the vaccine contains mRNA. The mRNA will be translated into a SARS-CoV-2 protein. The SARS-CoV-2 protein is an antigen that will trigger the immune response. So, ganun yung 3 types of vaccines natin. So, they're all effective, they're all safe. Hindi totoo yung sinasabi nila na, I mean, there are certain exceptions, pero as with any drug naman, lahat naman syempre merong possible side effects in very small portions of the population. And the population here is very large kasi halos buong mundo syempre nagbabakuna, di ba, for the COVID. So, pero all of them are helpful and all of them help us help end the pandemic. So si Sinovac, actually I was vaccinated with Sinovac and I was thankful na I was vaccinated with Sinovac kasi since the first vaccine that you got is the most helpful one, syempre. Yeah. Syempre, especially during that time na sobrang tindi pa ng COVID, hindi kagaya nung later eras na ng COVID, di ba, yung mga Omicron, wala nang ano. Pero yung mga unang phases ng COVID, sobrang tindi pa ng COVID vaccine bago siya naging parang flu na lang ngayon. So, the first vaccine that you could have gotten is the best one. So, I think, so sabi nila may mga studies na Sinovac, etc. is bad. Pero, personally, it helped save a lot of lives. So, and I'm thankful that we got these vaccines kahit pa paano. So, okay. So, next. Doon na tayo sa diagnosis-wise, very important to us as medtechs. So, you have your polymerase chain reaction. So, the polymerase chain reaction is a method for rapidly producing multiple copies of a DNA sequence. So, it has several uses. First of which is DNA fingerprinting, especially pag may gumawa ng krimen, tapos may naiwan siyang bakas dyan sa crime scene, they can use the DNA. They can use that very small sample they got from the crime scene and amplify that multiple times para ma-realize nila kung sino yung gumawa ng krimen. Okay? You can also use this for paternity testing, di ba? Baka cheek cells. Di ba madalas may ganun pag ipapanood nyo sa drama? Cheek cells, kukuha silang buhok or whatever. Tapos, ang litak na sample, pero pinapadami nila yung DNA contained in that sample using this polymerase chain reaction. Your PCR is also used in the diagnosis of bacterial and viral infections. Siyempre, pag nagpasa ka naman ng sample to the lab, 'di ba? Pag sinwab ka, 'di ba? Sinwab lang, sinungkit lang sa may ilong mo, sa may likod, sa may nasopharynx. Oh, ang liit lang ng sample nun, pero they were able to amplify that to make multiple copies of whatever's found in your nasopharynx para ma-realize kung meron ka bang COVID or wala, 'di ba? So next, and this also used for direct mutation testing. So para to dun sa mga may taong may genetic diseases. So what happens during polymerase chain reaction at bakit siya chain reaction? Kasi ito is a very important development na ginawa nung isang scientist. His name is Gary Mullis. He won a Nobel Prize for this, I think. Because it's very monumental and it has changed diagnosis as we know it forever once this was made. So what happens in polymerase chain reaction? You have your double-stranded DNA. So meron siyang 3 basic steps, pero it just happens over and over again until you reach the desired amount of DNA that you want. Okay. So you have your double-stranded DNA. What do? Your first step is denaturation. So how do you denature your DNA? You heat it up to approximately 95 degrees Celsius. Pag hinit up mo yung DNA to approximately 95 degrees Celsius, maghihiwalay yung strands niya. Okay? Next, what happens is, the next step is your annealing. So primers anneal to the sequence to be amplified. So magkakaroon ng primer. And DNA polymerase, yung tinatawag na Taq polymerase, which is something they discovered from an extreme environment, so malapit sa mga volcanoes, etc. So, 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 during annealing, you lower the temperature to 55 degrees Celsius, and then, primers are added as well as your nucleotides. So, your nucleotides mo will bind to the nitrogenous bases via complementary base pairing, and the DNA polymerase will link them together to form a new strand. Okay, so magkakaroon ka ng strand dyan, magkakaroon ka ng strand dyan. So, and then, you heat that up to 72 degrees Celsius again. So, during which your elongation occurs. So, your DNA polymerase will attach the DNTPs in the solution to the strand para maging binded na siya. Okay? Ulitin natin. So, first step is denaturation. Your double-stranded DNA is separated into 2 strands at 95 degrees Celsius. Next, during annealing, you cool it down to 55 degrees Celsius. And you add primers, you add DNTPs to the mixture. Okay? And the Taq polymerase. Okay? Next, during elongation, you heat it up again to 72 degrees Celsius. So, pag nakapag-anneal na yung DNTPs during the second step, ang gagawin ni DNA polymerase, it will attach the DNTPs to the strand. Okay? So, next. So, why is it a chain reaction? Kasi meron ka ng one strand here, you have another strand here. So, ang gagawin mo is you repeat the cycle over again. So, itong strand na to, hiinitin mo siya ulit at 95 degrees Celsius para maghiwalay. So, maghihiwalay ulit yung strand, maghihiwalay ulit yung strand. Dito din. Dito din. So, meron ka na ulit denatured na DNA strands, di ba? So, ulit. So here, meron kang pinaghiwalay na strand dyan, di ba? So, dito din, meron kang pinaghiwalay na strand. And then, after denaturation, you can then undergo annealing. You can then undergo elongation. So, anong mangyayari? Magkakaroon ka na ngayon ng, doon sa second cycle mo, you will now produce more DNA strands with each of the strands, di ba? So, ganun yung nangyari dito. So, from 2, you produce 4 DNA. So, kaya siya naging chain reaction. From 1, you produce 2. From 2, you produce 4. So, pataas ng pataas, padami ng padami. Kaya siya naging chain reaction using your polymerase. So, itong polymerase chain reaction is actually very monumental to diagnosis. Kasi ginagamit natin siya for diagnosis of many different diseases. Lalo na pag yung mga specialized na for example, 'di ba sa COVID naman medyo kakaiba kasi what we used in COVID is RT-PCR. So what happens in RT-PCR? It's reverse transcriptase PCR. So ang nangyayari doon, since your SARS-CoV-2 or yung COVID virus mo is an RNA molecule, you first convert it to DNA. So reverse transcriptase will convert the RNA of the SARS-CoV-2 into a DNA. And then yung DNA na yun will undergo the polymerase chain reaction para dumami siya. And then, pag marami ka ng sample, then you can test it for the uh, organism. Okay? So, I think that one is the last slide. So, thank you very much for listening to this lecture. I hope you learned something. So, we will have a quiz on this topic. So, make sure that you understood it. Okay? So, and I will see you next meeting.